Hello, dear friends and colleagues. On behalf of my colleague Sandrine Dubrac, I'm very happy to open today's ESDR Kitchen live webinar. You know, today we are in the category of freshly baked, and everybody knows that means high-class science and high-impact papers that are recently published by young ESDR friends and colleagues. So today we will have two exciting presentations, two very interesting manuscripts presented by the first authors of these manuscripts. And you know, this, as always, will be like 10-minute presentations directly followed after each other. And then we will have a little more than 10 minutes or so to discuss. So I would like to encourage you at this step to please ask your questions via the chat function to both Juliette and Yun-Tsan. Juliette Bertrand is the first speaker today, and you see she recently published a very interesting manuscript in the Journal of Investigative Dermatology. She will be followed by Yun-Tsan, who will be introduced by Sandrine in just a minute. So let's directly start with our first speaker today, and that is Dr. Juliette Bertrand. She did her PhD in molecular and cellular biology in Lionel Laroux's lab at the Institut Curie in Marseille, where she worked on what she will present today. And now she changed to a small startup and is the head of biology at this startup Skinosif. If that's correct, without making too much of an advertisement, Juliette, you can further elaborate that. And the title of your presentation is Loss of Dicers in Newborn Melanocytes Leads to Premature Hair Graying and Changes in Integrin Expression. When I first read it, I was very hopeful because I thought hair growing, but unfortunately it's hair graying. But you will enlighten us further. Please, Juliette, the floor is yours.
Thank you. So hello, everyone. I'm very happy to present today in the ESDR kitchen. So I'm Juliette Bertrand, as Kilian said. I'm working for a small company called Skinosif. It's developing innovation in sun protection. But today I'm going to present my PhD project that I did in Lionel Laroux's lab at the Institut Curie and was published recently in JAMA. So the pigmentation of hairs and skin is based on the presence of the pigment melanin, which is synthesized in melanocytes in organelles, which are called melanosomes. And these melanosomes are transferred to keratinocytes of the epidermis and of the bulb in hair follicles. So pigmentation is a complex process which is finely regulated by genetic, epigenetic, and environmental factors. So pigmentation alterations can be due to modification in melanocyte development, maintenance, and function. Here we see mainly two groups: genetic mutations, here, for example, that can induce hypopigmentation here, but also hyperpigmentation you see here, and environmental stress, which finally modulate pigmentation. For example, mechanical stress or UV. We focused on dicer, a protein whose activity is regulated by several stress. For example, in different cell types, its expression and activity are repressed by ROS, hypoxia, serum deprivation, phorbol esters, and interferon alpha. Dicer is a ribonuclease. It's involved in microRNA maturation. So without going too much in the details of the microRNA biogenesis, dicer is active in the cytoplasm here. It cuts a precursor hairpin microRNA here into a microRNA duplex, a double-strand RNA of about 22 nucleotides. Then dicer plays a crucial role in the regulation of many genes in the cell. So we wonder, is dicer involved in the fine regulation of pigmentation after birth? And so what would be the consequence of a lack of dicer in melanocytes? So to answer this question and to focus on the role of dicer after birth, we use an inducible dicer mouse mutant. On a C57 black background, we use the tier 3 ERT2 transgene, which express the Cray recombinase, which is spatio-temporally inducible by tamoxifen treatment. So we treated pups at birth by intragastric injection of tamoxifen at days 1, 2, and 3. This treatment induces the defloxing of dicer at birth in melanocyte stem cells and in melanocytes from the first hair cycle. This inducible mutant allows us to analyze the role of dicer in adult pigmentation. So, in dicer inducible mutant treated at birth, we observe hypopigmentation from the first hair cycle. After, we see increasing hypopigmentation with age. And for example, here at P60, we see some hypopigmentation, and this hypopigmentation is stronger on the belly than on the back. This phenotype is observed from birth, that the hypopigmentation is always stronger than on the belly than on the back. After quantification of the melanin content in hairs at this age, at P60, we conclude that the pigmentation of dicer mutant mice is significantly reduced both on the back and on the belly of dicer mutant mice compared to control mice. If we look closer, we can follow the distribution of melanin in hairs of mutant and control mice on the right. In wild-type mice, we see that the melanin is homogeneously distributed along the black hairs. And in mutant mice, we see that melanin can be homogeneously distributed in black hairs, but we also observe that some hairs are white. And if we look at the proportion of these white hairs, we see that we have more white hairs on the belly than on the back, which explains that the hypopigmentation is stronger on the belly than on the back. So hypopigmentation of dicer mutant corresponds to a salt and pepper phenotype, meaning a mix of white and black hairs. So in mice, hair pigmentation relies on melanocyte maintenance over hair cycle. In pups, the embryonic hair cycle is lasting for about 3 weeks, and then adult hair cycle starts with anagen, which is the growing phase. So melanocyte stem cells go out of quiescence and will give transient amplifying cells, which will migrate following the follicle growth and differentiate into mature melanocytes in the bulb. Here they transfer melanin to keratinocytes, which will give the pigmented hair. So to analyze melanocyte maintenance in dicer mutant, we looked at their localization on hair follicle section by DCT immunostaining. In dicer inducible mutant, most of dorsal hair are black, and DCT positive cells are found at the bulge and bulb, like in A, but can be mislocated as shown here in B. Okay. Most of dicer inducible knockout ventral hairs were white, and we observed either some DCT positive cells mislocated, like in C, here we see close to the bulge, in D and E, where they are at the periphery of the follicle, or also in some follicle they are absent, like in F. This observation suggests that in the absence of dicer, hair follicles would progressively lose their melanocytes. So melanocytes may undergo cell death or have a default in migration. So to understand the molecular mechanism of melanocyte loss, we use the transcriptomic approach in vitro. We use mouse melanocyte melanin cells in culture. They were transiently transfected with the siRNA against dicer or siRNA control and lyse 24 hours later. Total RNA was extracted for transcriptome analysis by microarray, and we had 280 transcripts which were deregulated in SI dicer compared to SI control. And among these 280 transcripts, we had 4 integrins which were upregulated when dicer expression level is reduced. We focus more specifically on alpha-V integrin. Its protein level is increased in dicer cells in vitro and also in vivo in the melanocytes here from the hair follicle of dicer mutant mice. So a Myriam analysis was done to decipher which microRNA regulates alpha-V integrin. And we focused on the MIR92B here, which was downregulated in dicer cells in vitro. To validate the target of MIR92B, a microRNA mimic was transfected in melanese cells, and we observed that alpha-V integrin RNA and protein levels were downregulated. We also repressed the stability of the luciferase RNA containing the MIR92B putative binding site from the 3' nuclear sequence of alpha-V integrin here. So it's downregulated, but when we mutate this sequence, we see that there is no repression indicating that MIR92B targets specifically alpha-V integrin in mouse melanocytes. So we investigated the role of alpha-V integrin dicer and MIR92B in regulating melanocyte migration by analyzing single-cell migration on non-coated plastic dish. In melanese cells, the reduction of alpha-V integrin with the siRNA reduced cell velocity. Then both the reduction of dicer with siRNA and the transfection of the antagomer for MIR92B increased cell velocity. So altogether, this result in identifying the dicer MIR92B alpha-V integrin axis as a signaling pathway affecting melanocyte migration. And through this pathway, dicer regulates melanocyte position and renewal in the hair follicle and participate to the hair pigmentation in adult mice. So I thank all the people in the lab who worked with me during my PhD and who collaborate with to this project, and collaborators also work on this project. And I thank you for your attention and be happy to take questions in the Q&A part. Thank you.
Thank you, Juliette, for this outstanding presentation. I remind people that if you want to ask questions to Juliette, to use the chat part. So now we are going to switch to the next speaker, Yun-Tsan Chang. So Yun-Tsan did his PhD in immunology at the Department of Dermatology of Zurich under the guidance of Emanuela Genova. Then he did about a 1-year postdoctoral fellowship in the same department, and then I guess followed Emanuela to Lausanne in the Department of Dermatology for a position as a research scientist. So now Yun-Tsan is going to present a work that was really nicely published in Nature Communication this year, which is entitled MHC-I Regulation. Can I have the title, please? Yeah, thank you. MHC-I upregulation safeguards neoplastic T cells in the skin against NK cells mediated eradication in mycosis fungoides. Yun-Tsan, the stage is yours for your presentation. Thank you very much.
Yeah, thank you very much for the nice introduction. And today I'm going to share with you our latest publication about the immune escape mechanism from tumor cell surface antigen target therapy in cutaneous T-cell lymphoma. Cutaneous T-cell lymphomas are a group of non-Hodgkin lymphoma, and they are considered as the malignancy of skin homing T cells. And mycosis fungoides is the most common type of cutaneous T-cell lymphoma, and accounting for about 50% of all cutaneous T-cell lymphoma cases. Impairment of cellular immunity is the major problem for cancer therapy. And with the development of antibodies targeting tumor cell surface antigen, we witnessed remarkable advances in treating blood cancers. However, the current challenge is that many tumors of hematopoietic origin in the skin remain resistant. Here you can appreciate large clinical observations about two therapeutic antibodies, anti-CD55 and also anti-CCR4 in treating cutaneous T-cell lymphoma. As compared to blood, you can find a low response rate in the skin, indicating that the skin tumor T cells are resistant to the therapy. So in this study, we used a causality supervised deep learning approach to identify the problems. So first, we isolate the skin cells from MF skin lesions, and we obtained in a total number of 17 tumor skin lesions from 14 MF patients. And afterwards, we purified the CD3 positive and CD45 positive T cells from the skin, and also sequenced them on a single cell level. The next step, to be able to separate the tumor cell and non-tumor cell, we reconstruct the natural T cell identifier, the T cell receptor, to be able to separate them. And the T cells share the same T cell receptor, and they are considered as clonal malignant skin T cells. And this already provides the basis for machine learning and also the artificial neural network, followed by model training, validation, and testing. At the end, we identify 48 important tumor-related genes. So to have a first magnetic insight of those genes, we perform a gene set enrich analysis, and two pathways has been identified. The first is the MHC class 1 pathway, and the other one is the negative regulation of natural killer cell mediated immunity. So first, we check the expression of MHC class 1 on different type of T cells, including the malignant T cells from the skin and also from the blood. And in addition, we also check the expression on the other type of skin cancers. And interestingly, we found that the upregulation of MHC class 1 is clear and highly significant in the MF malignant clonal T cells as compared to other type of T cells. And also, it's significant highly expressed compared to other type of skin cancers. So what is MHC class 1? I think people know more about the function of antigen presentation, but it's also have another function for to provide the inhibitory signal for NK cell, and this function is very important for self and non-self recognition. So MHC class 1 recognized by the KIR receptor, which provide a very strong inhibitory signal for NK cell. And NK cell is the key effector cell for therapeutic antibody, and antibody-dependent cellular cytotoxicity, abbreviated as ADCC, is the main mode of action of this kind of therapy. So if the tumor cell highly express MHC class 1, then they will provide a very strong inhibitory signal for NK cell, and afterwards they inhibit the ADCC activity by NK cells. And indeed, we found that autologous skin NK cell can trigger effective ADCC activity against healthy skin T cells and also healthy bystander skin T cells. But the skin NK cell cannot mount effective ADCC against malignant skin T cells. To prove the relevance of our findings, we try to block the interaction between MHC class 1 and KIR receptor. And here we use the MHC class 1 blocking antibodies. And as expected, when we block the inhibitory interaction, we increase the ADCC we increase the ADCC activity against the malignant skin T cells. However, MHC class 1 is as I mentioned before, it's a very important molecule for antigen presentation. So directly block MHC class 1 cannot be used in real life settings. So to avoid this problem, instead of targeting to MHC class 1, we target to the KIR receptor on the NK cell side. Try the same to try to block the interaction between MHC class 1 and also KIR receptor. And here we use two different kind of therapeutic antibody, either by anti-CD55 and or anti-CCR4. Both antibodies show significant restore ADCC activity while the KIR receptor was blocked. At the end, we also want to confirm this finding in vivo mouse settings. So here we use the EL4 cell line. It's a marine T-cell lymphoma cell line. And I would like to mention that H2 is the marine MHC class 1 molecule, and LY49 is the functionally analogous to human KIR receptor. So here we have EL4 cell line, and this EL4 cell line was engineered to express human CD20, which allow us to use as the target antigens. And we also use the LY49 blocking antibody and try to have the settings. So we've already confirmed that EL4 cell line highly express MHC class 1 as the same as MF skin T cells. And also we confirmed the expression of human CD20 on EL4 cells. So here we have 5 different treatment. So treatment group, the two control group, either we treat with PBS or the isotype antibodies. And the other 2, we have the monotherapy, either treat with a therapeutic anti-CD20 antibody or the blocking anti-LY49 blocking antibody. And at the end, we have the combined therapy. And as a result, you can appreciate on the right, the combined therapy significantly reduced the tumor growth and also significantly increased the survival rate. So to sum up of this study, we used a connectivity supervised machine learning approach to identify MHC class 1 highly expressed on MF skin tumor T cells. And with the therapeutic antibody, if you have more MHC class 1, they will create a lot of inhibitory signal. So at the end, you have decreased ADCC activity, which may cause the treatment failure. And with the with the therapeutic antibody together with the blocking antibody, you can block the inhibitory signal for NK cells, so result in increased ADCC activity and enhance the treatment. So here I would like to thank my supervisor, Professor Emanuela Genova, and also the group members, and also all our collaborators from Linz and also from the Europe. And also thanks for the funding agencies to support these studies. And also thank you for your attention.
Thank you very much for this excellent presentation. And now the 2 papers are open for discussions. So please write your question in the chat part. There is actually a lot of questions from one person to Juliette. So the person is writing that it's a very nice presentation. And the person has different questions. So for example, Juliette, do you have an explanation why you have more gray hair on the belly than in the back? If there is this hair loss related to human diseases or just to hair growing? Yeah. And if this hair only in hair follicle melanocytes or also in skin residing melanocytes? And if so, is it involved in any skin diseases? So Juliette, yeah,
Okay.
if you can answer all these questions.
For the first question is about on the stronger hypopigmentation we have on the belly than on the back. We think it's just because there are less melanocytes on the belly, and so just the effect is stronger because it's more diluted. In the back, you have more melanocyte to support, so the effect is strong, the less strong. For the skin, yeah, sorry.
No, if just go on. Sorry.
Yeah, I was thinking in which where to start. About the skin and hairs. So we know that melanocyte migration is very important for pigmentation of hairs because the differentiating melanocyte, so the TAC, will migrate to the bulb to give the pigment to the hair in the bulb. So for this step, the migration of melanocyte is very important. So this mechanism we observe is important for pigmentation of the hair. I'm not sure it is. I really don't have the answer because we did not study the effect in skin pigmentation. But I think maybe the migration of melanocytes is less important, even though the interaction with the extracellular matrix might be an important point. And in this case, integrins might be important to have a good localization at the basal membrane for melanocytes. So it might be important, but we have not analyzed this. I'm not sure. And so we have not linked this hair graying to disease, but I would say as dicer is regulated by stress, it might be something that happens when you have hair graying induced by stress. So either mechanical stress or like, for example, we see in vitiligo when you have some loss of pigmentation and you lose actually melanocytes. So dicer may be involved in this case in response to stress and in the hypopigmentation. So skin disease.
Now we can tell everybody, right, Sandrine, that we already have the dicer containing shampoo.
Definitely.
Your startup will do something here, Juliette. May I ask Yun-Tsan, because I really think it was an outstanding presentation, very nice data, thanks a lot. Two questions, brief questions to you. Were there differences in the level of MHC-I in between the different MF samples that you chose, or do you think it's a very homogeneous level of MHC? It was highly upregulated in all MFs. And the second question would be, just answer.
Yeah, so we've already tested like more than 10 samples, and because we use the T cell receptors where we can separate the tumor cell and non-tumor cells. And it's quite, so the tumor cell is always upregulation of MHC class 1, but some samples that they upregulate more than the others. Yeah, but we all see this upregulation in samples, yeah.
And you trace it nicely back to NK cells. Do you see a role for CD8 positive killer T cells and adaptive immunity here also, or not yet investigated?
Yeah. I think when I present this, I always receive what about the CD8 positive T cell, because the tumor cell highly express MHC class 1, which may increase the antigen presenting to CD8 cells. But so far we have some ideas, but it's still not clear. About this. We found that the MHC although it's highly upregulated in a tumor cell, but when they contact with CD8 cells, it seems to be a little bit less expression during from the contact site. And until now, we don't have the explanation for that, but maybe further experiment will be needed. Yeah.
Thank you very much for these answers. Maybe it's time to move on and to close this webinar. Again, thank you very much for the presenters who did really an excellent job, and for all people who listened to this webinar. So now we remind you that you can have access to the webinar on the ESDR Kitchen webcast. It's available if you want to watch again this webinar, if you want to watch the previous ones. So we invite you to go there. I would like to advertise for the next ESDR Kitchen session with a topic of skin aging and senescence. And the two presenters will be Peter de Keyser, who is really a specialist of senescence, and Peter Yun-Tsan Chang, who is really a specialist of skin aging, and really looking forward to these two presentations by two outstanding researchers. I remind you that this ESDR Kitchen episode will take place on Thursday, the 13th of June, and not on Wednesday. So please notice these changes in the schedule of the ESDR Kitchen. Also, very important, we really encourage you to attend the ESDR meeting in Lisbon that will take place early September. You are we will absolutely appreciate that you submit your work to this meeting. Abstract submission is until June 3rd. And for late-breaking abstract submission, it will be in the 1st and the 19th of July. Yeah, and so we can say that now the early fees registration is opened. So thank you very much for everybody, and see you next time. Thank you.